As you can see for this unit, the format of our slides is a little bit different. Fill in the blank, which is a lot easier for you. Hopefully it will be as effective. And at the risk of being boring, I'm basically just going to read through this with you. And of course, you'll be filling in the blanks on your copy. And I'll explain further when necessary. So let's get started here. We're looking at genes and variation. And if you'll recall, this unit has to do with populations and microevolution. And microevolution has everything to do with slight changes, small changes. That's where the micro comes in. So many genes have two forms or alleles. Some genes don't. Some genes only have one form, so there aren't two alleles. And if you'll recall, if there are more than two alleles, we refer to that as multiple alleles. For example, blood type, ABO blood type, has three alleles, the A allele, the B allele, which are codominant, and the O allele, which is recessive to both A and B. So things can get complicated with multiple alleles. And all organisms have genetic variation that is neutral, meaning that it's not affecting anything in an either positive or negative way. For example, you have mutations. We all have mutations, but they're not making any difference in the way that our bodies are operating or our metabolism is operating. So they are neutral. Another way of looking at it is they are neutral to our survival, so they're not affecting our survival either positively or negatively. And I don't know if you've thought about it or not, but you are heterozygous for many genes, which means that you carry alleles that are not being expressed. They're being masked by the dominant allele, and therefore you're not showing those characteristics or traits. So they are invisible. They're invisible because they're recessive. But they can very well show up in your offspring or in your grandchildren even. So as it says here, genetic variation is studied in populations. And in this photo, we see it's not really a population. It's just a group of ladybugs, but we could consider this a small population of ladybugs. And there is variation. If you look at the spots on the ladybugs' exoskeletons, they vary. They vary in number. They vary in size. So speaking of whether or not we could consider this a population, we need to define population if we're going to study variations in population. So a population is a group of individuals of the same species, and as I told you previously, a definition that I gave you, it's not only the same species, but it's the same species in the same place at the same time. So really there are three components to a population: same species, same place, same time. Another concept that's important in what we're studying now is the concept of a gene pool, which we've run across previously. And it consists of all the genes, including all the different alleles that are present in a population. So if this were our population, and we're talking about the genes that are responsible for the number of spots on the ladybugs, for example, and let's say that there are two alleles. So we're talking about each of those two alleles in each individual of this population. So we would count up the number of ladybugs and multiply it by 2, and that would be the number of alleles in the population. And that's what we're talking about. We're talking about the alleles in the population, present in the population, and you can specify that you're talking about the gene, just one gene, or you're talking about all the alleles of all the genes in a population. That would be the gene pool. Another example is how it came up previously. Again, with ABO blood type, there are three alleles, the A, the B, and the O, and one individual can only have two alleles, one from the mother and one from the father, but there are three alleles in the human gene pool. So out there in the human gene pool, in the whole human population, there are three alleles, and it's possible that you could get any two of those alleles. So if we consider a gene pool, and all the alleles, or the alleles for any one gene anyway in the gene pool, you can calculate what's known as relative frequency. The relative frequency of alleles, you can calculate the relative frequency of genotypes, and you can calculate the relative frequency of phenotypes in a population. But let's start with the allele. It's the number of times the allele occurs in a gene pool compared with the number of times other alleles for the same gene occur. So relative frequency is often expressed as a percentage, but it can also be expressed as a decimal. And when you're doing calculations, you want to definitely be using decimals, not percentages. So in this example, we're looking at the gene for fur color in mice, and black is dominant over brown. And so this represents all the alleles in the population. So all the brown alleles, all the black alleles. And if we count them up, the total number of alleles is 30. But if we count up the number of brown alleles, it's 50. Yeah, my bad. The total is 50. But if we count up the number of brown alleles, that's 30. So 30 divided by 50 is 0.6. And so that's how I prefer to leave the allele frequency. I like to talk about allele frequencies expressed as a decimal instead of as a percentage. But when it comes to genotypic frequencies and phenotypic frequencies, those I like to talk about as percentages. So if the brown allele frequency is 0.6, that means that even without calculating it, you would know that the black fur allele is 0.4, because 0.6 and 0.4 has to add up to 1, right? And that's where relative comes from. We're talking about the relative frequency, and what makes it relative is they're being compared to each other, and together they total the total, which is 50, or 1. Now, the only way we can calculate the relative frequency of alleles here is because we can see all the alleles. In this example, in this diagram, we can see them. Normally, you can't see them. So what you normally have to do is you have to calculate the allele frequencies from the phenotypic and genotypic frequencies. In other words, go backwards. Here we're going forwards in this example. So we're going from allele frequencies to genotypic frequencies. So let's start with the brown allele. So the brown allele frequency is 0.6, and we can calculate the percentage of the brown furred individuals in the population simply by multiplying 0.6 by 0.6 to get 0.36, and then multiply by 100, and that's 36%. So we can expect 36% of the population to have brown fur. We can expect 16% to have homozygous black fur. So that would be the allele frequency for the black allele, 0.4 times 0.4 to represent homozygous, and that's 0.6 multiplied by 116%. And when it comes to the relative frequency of heterozygous black mice, it would be the black allele the black allele frequency times the brown allele frequency, which would give us 0.24. And then we multiply by 2 because, if you'll recall from the Punnett square, there are 2 separate combinations of gametes that result in the heterozygous individuals in a population. So we have to multiply by 2, and that gives us 0.48. Multiplied by 100 is 48%. And being able to calculate the homozygous dominants and the heterozygous individuals in a population is a real useful thing to be able to do, because normally, you look at the phenotypes here and you can't tell these mice apart. They're both black, whether they be homozygous black or heterozygous black. They're both black, and you can't see their genes. But we can still use allele frequencies to calculate the expected percentage of the population that is heterozygous or homozygous. And that can be useful, especially if you're talking about a genetic disorder. So let's think of a situation where maybe the environment changes and it would cause natural selection to take place, and that would result in a change in the allele frequency in the population. Let's say, for example, there's a volcanic eruption, and so these mice are now living on black rock because, lava, when it cools, it forms basalt, which is black. So that would mean that the brown mice would stand out more against the black environment, and birds would come along, like hawks and eagles would come along and be able to pick off the brown mice more easily. So predation would remove more of the brown mice from the population. And that means the brown alleles are being removed from the population. So the allele frequency would change. The allele frequency, the brown allele would decrease. And that's evolution. I mean, even if we were to take one of these brown alleles and turn it to a black allele, it represents mutation, or that represents evolution. It's very, very tiny evolution. That's why we call it microevolution, but it's evolution nevertheless. So in genetic terms, evolution is any change in the relative frequency of alleles in a population. Any change, no matter how small. And that's why it's referred to as microevolution. So where does genetic variation come from? Well, it ultimately starts as mutation, but mutation is relatively rare. And mutations don't always affect the organism's phenotype. Therefore, those changes are not acted upon by natural selection. So, but all differences between individuals in a population started out as mutations. However, most of the change or variation between individuals in a population comes from sexual reproduction and gene shuffling that takes place during sexual reproduction. So gene shuffling or recombination, like what takes place during meiosis and the production of gametes, during prophase, crossing over takes place, and that represents shuffling of genes. And as I've told you before, that's the rule, not the exception. Crossing over happens all the time. And that produces recombinant individuals in the next generation. They don't look like their parents. And that's where most variation comes from. But all sexual reproduction can do is shuffle the genes. So it doesn't change the frequency of alleles. All it can do is remix them and shuffle them. Just like you have a deck of cards, 52 cards, you shuffle them. It doesn't change the number of cards, and it doesn't change the cards themselves, but it does change the combination of those cards. And again, crossing over increases the number of genotypes that can appear in offspring, meaning that the offspring are not all parental types. There are also recombinants. So most human traits are not single gene traits, and single gene traits are what we would consider inheritance by simple Mendelian means, because that's all Mendel dealt with. All 7 characters of his pea plants that he studied were controlled by single genes. But it turns out the number of phenotypes produced for a given trait depends on how many genes control the trait. So there are single gene traits that control human traits, but most human traits are polygenic. But we're looking at single gene traits first here, so and that's what you're most familiar with, because that's what we've been dealing with most of the time. But a single gene trait is controlled by one gene that has only 2 alleles. However, there may be multiple alleles, and that would still be considered a single gene trait. Variation in the gene leads to 2 possible phenotypes, unless you're dealing with incomplete dominance, for example, then there might be 3 phenotypes. And again, if you're dealing with multiple alleles, there can be more than 2 possible phenotypes. But in simple Mendelian terms, 2 phenotypes, like in this case, widow's peak. And widow's peak is dominant to not having widow's peak. However, the presence of widow's peak is less common in the population, and that may seem strange, but if you, again, have the concept of gene pool and the idea that in the human gene pool, there really aren't that many dominant alleles for widow's peak. In other words, most of us are, well, if we don't have a widow's peak, that means we're homozygous recessive for not having that widow's peak. And there aren't that many dominant alleles out there in the human gene pool, so it's less likely that you're going to get one of those dominant alleles. So phenotypically, having a widow's peak is less common in the human population because there aren't that many dominant alleles for widow's peak out in the gene pool for a person to inherit. So just being dominant doesn't necessarily mean that that's going to be the most common characteristic in the population. It's also all about the frequency of alleles. Like I pointed out, many human traits are controlled by more than 2 genes, and therefore they're polygenic traits. And polygenic traits don't just have 2 phenotypes, like tall and short. in pea plants, that would be tall and short, is what I'm referring to. One polygenic trait can have many genotypes and phenotypes because there are a number of different genes that are interacting together. So height in humans is a polygenic trait. So here we're looking at one of the tallest men, and I think, this is an old image. So the tallest man in the world is probably not this guy anymore, and the smallest man in the world is probably not this guy anymore, but it shows the extremes of height, which is our example here. So height is a polygenic trait, and you can tell by looking at the distribution of that trait in the population whether it's a polygenic trait, because if it is, it's going to form a bell-shaped curve. There's going to be a wide range of phenotypes, and when those phenotypes are graphed, they form normal distribution, or also known as a bell-shaped curve. So very few people are very short, and very few people are very tall. Most people are somewhere in between, and there's a lot of variation in between, because it's a polygenic trait. There are many genes that are interacting to produce that trait. So what's the connection between this concept of allele frequency and gene pools to evolution? Well, natural selection affects which individuals survive and reproduce and which do not. And if an individual dies without reproducing, it doesn't contribute its alleles to the population's gene pool. In other words, its alleles are removed from the gene pool, and that means that the relative frequency of those alleles changes, and in this case, decreases. On the flip side, if an individual produces many offspring, and if you'll recall, that's Darwin's concept of fitness. So individuals that produce more offspring are more fit. Then its alleles stay in the gene pool and may even increase in frequency. So the idea here is to try to get across to you the definition of evolution, in that it is any change in the relative frequency of alleles in a population from generation to generation. And there is almost always some change in the frequency of alleles in the gene pool of a population from generation to generation. Therefore, evolution, albeit microevolution, is always happening. And the question here, can you calculate the relative frequencies? Well, I'm just going to describe how you would do it. So if you wanted to calculate the frequency of the recessive alleles, just count up all the lowercase b's and then divide by the total number of alleles that you can count, not only here, but in the individuals in this diagram. In this diagram, we're looking at mouse coat color again, and what's being shown down below here represents the relative frequency of the alleles of those mice. And it kind of does a good representation of showing that it's relative, and that there are fewer recessive alleles than there are the dominant alleles for darker fur. And one last time, evolution is any change in the relative frequencies of alleles in a population, no matter what caused that change. So in this example, immigration, in other words, beetles flying into and joining a population, is going to bring in alleles that weren't in that population's gene pool before. So immigration, emigration, mutation can change the allele frequencies of a population, and most importantly, natural selection can change the allele frequencies in a population, and that's microevolution.